It is a pleasure and an honor to participate in this Darwin Day 2023 program hosted by the Tardid School of Philosophy. I'm excited to share the virtual stage with such a remarkable lineup of scholars, to celebrate the work of Charles Darwin, and to share my thoughts with all of you on natural selection and the origin of form. What I wish to consider is a question about a central claim of Darwin's. At the end of The Origin, one finds the following famous passage. It is interesting to contemplate a tangled bank, clothed with many plants of many kinds, with birds singing on the bushes, with various insects flitting about, and with worms crawling through the damp earth, and to reflect that these elaborately constructed forms, so different from each other and dependent upon each other in so complex a manner, have all been produced by laws acting around us. These laws, taken in the largest sense, being growth with reproduction, inheritance, which is almost implied by reproduction, variability from the indirect and direct action of the conditions of life, and from use and disuse. A ratio of increase so high as to lead to a struggle for life, and as a consequence to natural selection, entailing divergence of character and the extinction of less improved forms. It is here that Darwin makes a bold claim about the consequences of natural selection, namely that such a process entails the divergence of character and thus production of life's variety. My question is simple. Is this true? Does evolution by natural selection account for the diversity of living things we see around us? Of course, as asked, this question is vague. There are many ways to make it precise, many aspects of the diversity of living forms that we might reasonably expect a theory of the origin of species to answer. We might, for example, expect such a theory to predict a rough order of magnitude for the number of species extant at any one time, or to tell us how often new species should be produced. I will focus here on the relationship between two sorts of diversity. One sort is taxonomic, the number of species or varieties, or in other words, the number of distinct clusters in morphological space. Many distinct clusters of form, however, does not entail a wide divergence of those forms. For the novice birder, at least this novice bird watcher, sparrows are ridiculously hard to tell apart because the species in my area all look more or less the same. Not so, however, for the Membracidae, the family of insects comprising the tree hoppers. As these 5 examples, all from Paraguay, illustrate, the species of this family fill in a large volume of morphospace. That is, they exhibit wildly different forms. This is morphological disparity. Both taxonomic diversity and morphological disparity can be quantified. We can simply count the number of species, and there are many ways of capturing differences in shape or form. For ostracods, a big class of small crustaceans that left plenty of seed-shaped shells in the fossil record, we can observe how both sorts of diversity have changed over geological time. Wan et al., for instance, focused on the tumultuous period of time known as the Permian-Triassic extinction, the largest known mass extinction in which some 87% of marine invertebrate genera went extinct. Over that interval, both the diversity and disparity, as exhibited by shell morphology, of ostracods changed, as one might expect. However, this change was not uniform across global ostracod assemblages. In most locales, both taxonomic diversity and morphological diversity dropped in lockstep over the interval of the extinction event. But in some parts of the world, represented in the chart on the right by formations in present-day China and Turkey, species-level diversity actually increased while morphological disparity either remained the same or even plummeted. In a classic paper by Mike Foote, the two sorts of diversity were tracked over a much longer span of time and place, encompassing the entire Ordovician, Silurian, and Devonian periods and about half of all known species of the clade in question, namely the crinoids. Crinoids comprise a class of echinoderms known commonly as sea lilies. These marine animals are mostly stalked, sessile creatures with a crown of feathered arms for passing food to a central mouth. Their bodies are made rigid by a large number of calcareous segments, which upon death scatter like beads and tend to leave characteristic ring-shaped impressions in the fossil record. For much of the Paleozoic, they were tremendously abundant. The two plots on the right show us how this abundance manifested in diversity over time. The upper plot depicts a quantification of morphological disparity, which, after rapidly reaching some maximal value in the earliest radiation of crinoids, remained more or less constant over the millennia. By contrast, taxonomic diversity rose steadily until the end of the Devonian. The evolutionary history of these two groups forces us to confront a puzzling discordance. In the ostracods, we see that sometimes taxonomic diversity increases while morphological disparity declines. In the crinoids, we see that morphological disparity can increase and saturate faster than taxonomic diversity in radiation events. So, why and when do diversity and disparity decouple? That is, why and when should we expect taxonomic diversity to change differently than morphological disparity? This is one very specific question we can reasonably expect any theory about the origin of the diversity of living forms to answer. I claim that evolution by natural selection does not entail any such answer. Of course, evolutionary biologists have explored this divergence of diversity at length, and there are in fact a range of explanations and approaches to explanation on offer. We find them in the literature on macroevolutionary modeling, that is, modeling of evolutionary change at the level of speciation and the emergence of higher taxa. At this scale, the elements under consideration are not individual organisms or even populations, but rather clades within extended lineages, and the volumes of morphospace, the abstract space of possible biological forms or morphologies that those clades can occupy. How are the changes in taxonomic diversity and morphological disparity modeled at this level? Let's consider one well-cited model by Sergei Gavrilets. This is surely not the final word on the matter, but it gives a sense for the sorts of biological features commonly brought to bear. This particular model is intended to capture the evolution of a monophyletic clade driven by extinction, speciation, and anagenetic changes. The variables invoked are listed here. Chief among these is n, the number of lineages in the clade being modeled, and a straightforward measure of taxonomic diversity, and d, a measure of morphological diversity applicable to a clade and characterizing the extent to which it fills morphospace. The variables sigma 1 and sigma 2 are probabilities, specifically the probability of speciation with and without morphological change respectively. There are two important things to emphasize here. The first is that no attempt is made to explicitly represent the process of speciation. Rather, it is assumed that over the timescales of interest, the outcome of this process is captured by the given pair of probabilities. Second, these probabilities pertain to a single clade. Insofar as this model can be made to capture the actual rates of change in real clades, as opposed to broad patterns of evolution, these probabilities reflect information about the nature and history of the particular organisms in this clade. Similarly, both population and ecological information is captured by an additional pair of probabilities, delta 1 and delta 2, representing the probability of a lineage going extinct on its own or as part of a subclade respectively. The overall rate of extinction is just the sum of these two probabilities. All of the vast ecological complexity concerning the interaction of organisms, their ways of life, the environments they occupy and influence, and the dynamics of interacting populations of predators, prey, and symbiotes are all elided in favor of these gross averages. The details of the complex link between heritable genetic factors and morphology are invisible at this level of description, and represented instead by a pair of probabilities, mu1 and mu2, that a given trait, assumed to be discrete, changes state. They reflect trends in motion through morphological space. Put together, these high-level descriptions of a clade allow for the representation of change in taxonomic diversity and morphological disparity as a set of difference equations. Specifically, the change in diversity is given by the difference between speciation and extinction rates times the current number of species, while the change in disparity is given by the difference between the intrinsic rate of morphological change and the diversity of form lost to extinction and to speciation without significant morphological innovation. Importantly, these changes can take on opposite signs, with one sort of diversity increasing while the other decreases. For completeness, I've also included the equation for little d, the net morphological evolution of an entire clade relative to an ancestor. But neither this additional equation nor the details of the Gavrelet's model concern us here. What matters is the general form of a solution to our macroevolutionary question that is exemplified by the Gavrelet's model. Any such solution will posit a relation among 5 sorts of variables. First and most obviously are measures of both taxonomic and morphological diversity for the clades under consideration. Second, we need a pair of functions from clade state and history to probability distributions that indicate how a clade will likely gain or shed taxa and how likely it is to expand or contract in occupation of morphological space. Finally, we need a set of variables that determine the mapping from clade history to each of the aforementioned probability distributions if these are not constant across time and clades. In other words, we need a theory of what determines the sensitivity of a particular clade to its history and present ecological state. It is presumably here that an explanation of diversity of either sort lies. To return to our original question, can the theory of natural selection fill this gap? Does natural selection ultimately provide the connection between history and ecological context and diversification? An answer depends, of course, on what we take natural selection to be. Darwin himself said that this preservation of favorable individual differences and variations and the destruction of those which are injurious I have called natural selection. A pithy summary from the modern literature asserts that, quote, natural selection is defined as variability among phenotypes in fitness. With a little more care, we can borrow Peter Godfrey Smith's necessary and sufficient conditions for a process of change in a population to constitute evolution by natural selection, or ENS. First, there must be variation in the characteristics of members of the population, which second causes different rates of reproduction, and finally, which is inherited. I'll adopt the following more compact formulation. ENS is a type of process in which different rates of reproduction among types within a population are cyclically caused by heritable traits that vary among the types. With this clarification, let us return to our central question, which we can now frame this way. Given the history of a clade and its current ecological context, does the fact that a lineage is undergoing ENS imply that disparity and diversity will or will not diverge? Of course, as we've already established, ENS refers to entities at the wrong spatial and temporal scale. So what we're really asking is whether ENS entails outcomes at these low-level scales that account for the high-level pattern of interest. If it's going to do the job, we need ENS to entail probability distributions over speciation, extinction, and morphological change for particular lineages. The mathematical theory of ENS is at present generally taken to be population genetics. Here is a plain vanilla model of the theory. The fundamental quantities tracked are the relative frequencies of alleles, variants of a gene, within a population. In this case, there are 2 alleles whose relative frequencies are indicated by P and Q. In the simplest case of non-overlapping generations, these quantities can be tracked through time with a simple difference equation, such that the frequency of an allele in generation t plus 1 is some function of the frequencies of alleles at generation t. That function is parameterized by the fitnesses of the 3 possible genotypes of a diploid organism with 2 alleles, denoted here as W11, W12, and W22. These fitness parameters reflect all of the salient facts about survival and reproduction for each genotype in the environment in which the modeled population is evolving. Right away, however, we are confronted with an apparent impasse. As Bruce Gleamore has pointed out, those fitnesses, the drivers of change in ENS, are exogenous to population genetics models. Why is this a problem? Because we need ENS to tell us how a given lineage is likely to change and split given its history and ecological context. but all of the latter facts are packed implicitly into the fitness parameters. It seems that the very thing we need as an output of the theory must be provided as an input. There are at least three significant obstacles to making this connection, making the values of the fitnesses endogenous to the theory of ENS. First, there is no direct connection between changes in gene frequencies and changes in morphologies. Second, there is no explicit connection between fitness and ecological variables. And third, modeling long-term change requires an exogenous mutation function. In a rebuttal to Gleamore, Peter Gildenhuys pointed out that in general the influence of environmental variables and changing conditions can be captured by population genetic models by partitioning populations and deploying fitness functions. While this is certainly true, it doesn't resolve our impasse. It is this very permissiveness of population genetic models and the theory of natural selection they're meant to embody that is the problem. With no restriction on the function from p and q at a time t to p and q at a time t plus 1, any set of causes can be accommodated. Conversely, no set of causes is implied. That means we must still attend to the specifics of each and every population to determine even the basic contours of an appropriate selective model. This can certainly be done, and productively so. To borrow an example of Bruce Gleamore's, Peter van Tienderen advocates for a particular mathematical framework called elasticity analysis for coupling demographic models of population change with population genetic models of selection. The details of so-called elasticities do not concern us here. What matters is captured by Figure 1 of that paper and shown here. The explicit coupling of traits with components of fitness like survival and reproduction rates in a specific environmental context. In other words, if we can model the causal details that determine how organism traits are likely to fare in a particular environment, then our models can actually predict particular shifts or probabilities of shifts in trait distributions. Of course, this works only for a snapshot in time at which we have observational and experimental access to the relevant causal relationships. Establishing the causal interactions between organism traits and the properties of their ecological context responsible for components of fitness requires the sort of fine-grained observation acquired at great expense that made the work of Peter and Rosemary Grant on Darwin's finches famous. In the ideal limit, at least in the absence of a causal theory at a higher level of description, one would attend to the salient interactions of individual organisms with their environment. One such study by Woodgate et al. Produced one of my favorite scientific figures depicting individual foraging sorties by individually identified bumblebees. Each panel is a visual summary of a little lifetime, and each is a perfect icon representing the kind of information we need to average over in order to connect ecological context to population change in models of natural selection. What detailed work like this confirms is that, as ENS suggests, organism traits are causes of their differential reproductive success. There is always a causal history for an individual organism, for a colony, a population, a lineage. What is unclear and left unanswered by the theory of ENS is what, if anything, unites these histories under a single type of process. This leaves us with a dilemma. Either evolution by natural selection cannot connect the relevant variables for anticipating changes in diversity and morphological disparity because all of the salient facts are exogenous to models of ENS, or we build models that can generate the right sort of low-level connections, but those models make no essential reference to a distinct and unifying process of ENS. In summary, then, the argument is this. Suppose that the salient facts about fitnesses and mutation are ineluctably exogenous to ENS. Then ENS puts no constraints on the relations among low-level properties of organisms or populations and their ecological context that could provide the probabilities needed for macroevolutionary models. Suppose that we focus instead on causal models that can generate the right sort of low-level predictions for individual populations in specific ecosystems and attempt to generalize from them. Those models make no essential reference to a distinct process of ENS. Either way, ENS is an irrelevant way of describing the low-level processes that aggregate to produce macroevolutionary change. That is, ENS is irrelevant to predicting the relationship between morphological and taxonomic diversity. ENS is not false, nor is it vacuous. It's merely impotent for answering questions about the origin of biological diversity of the sort we've been considering. Consider, if you will, the following osmotic analogy. Suppose we're trying to predict what will happen to two solutions separated by a semi-permeable membrane that differentially restricts diffusion depending on the type of molecule, allowing little a solvent molecules to pass, but blocking big A solutes. On the one hand, we might entertain a theory of chemical selection. Such a theory posits that each type of molecule has a fitness for crossing the membrane to the right. Over time, molecular types with greater rightward fitness increase in relative frequency and absolute abundance on the right. The fitness of a molecular type is a function of the environment, that is, of the nature of the semipermeable membrane and of the frequencies of all types of molecule present in the solution. On the other hand, we might consider a chemical potential model. The general theory of statistical thermodynamics tells us how mixtures of m different chemical species must behave. Specifically, it relates differential changes in internal energy to changes in entropy, pressure, and the numbers of chemical species. When given the particulars of the environment, this equation entails an equilibrium concentration on both sides of the membrane. At low solute concentrations, where the Van 't Hoff equation applies, the proportion of solvent or a type molecules on the right is given by equation 6, where n sub AR is the number of big A solute molecules on the right, and n sub AL is the number on the left. We can ask of these two models, what is exogenous? For the selection model, the answer is just about everything. Most importantly, even the form of the dependence between fitnesses, the environment, and the frequencies of molecular types. It's clearly possible in this case to do better. The chemical potential model requires only information on the environment and the initial distribution of molecular types. Clearly, in the case of biological evolution, we lack an analog of the thermodynamic theory of chemical potential. We have not identified any regularities of any significant generality for the relation between ecological contexts and specific organism traits that would allow the theory of ENS to provide the kinds of facts necessary to answer our higher level question about the origin of form. Nor do we have any such theory at that higher level that directly couples a clade's ecological context and history to its likely changes in disparity and diversity. So how should we resolve this impasse? How could we answer the question that ENS does not? I'm no Darwin, and I have no such theory to describe, but I can suggest three broad lines of approach. First, we should embrace direct causal analyses at the lowest levels of description of the interaction between populations of organisms and their environment. It's here that we will find the facts that can sustain the right sort of generalizations to explain the tendencies of entire clades. That is, let's learn as many specific models as we can that actually predict the shift in trait distributions endogenously. Second, we should focus on generators of form, not constraints on selection. I've spoken little about mutation in this talk, but no model of the evolution of morphological disparity can be had without a positive characterization of the way lineages can move through morphospace. And finally, we should continue to seek fresh theories about the connection between low-level causal processes in populations and robust macroevolutionary and macroecological models. We know ENS doesn't fill this gap. Rather than continuing to offer post hoc descriptions of lineage evolution as the outcome of selective processes, we should keep an open mind about the form of the missing biological generalizations. Darwin's work on the origin of form framed a grand and exceedingly difficult scientific problem. we should honor that work by redoubling efforts to complete it. Thank you all for your attention, and I look forward to your comments.